The Wnt signaling pathways are a group of signal transduction pathways made of proteins that pass signals from outside of a cell through cell surface receptors to the inside of the cell. 3 Wnt signaling pathways have been characterized: the canonical Wnt pathway, the non-canonical planar cell polarity pathway, and the non-canonical Wnt calcium pathway. All 3 Wnt signaling pathways are activated by the binding of a Wnt-protein ligand to a Frizzled family receptor, which passes the biological signal to the protein Dishevelled inside the cell. The canonical Wnt pathway leads to regulation of gene transcription. The non-canonical planar cell polarity pathway regulates the cytoskeleton that is responsible for the shape of the cell, and the non-canonical Wnt calcium pathway regulates calcium inside the cell. Wnt signaling pathways use either nearby cell-cell communication or same-cell communication. They are highly evolutionarily conserved, which means they are similar across many species from fruit flies to humans. Wnt signaling was first identified for its role in carcinogenesis, but has since been recognized for its function in embryonic development. The embryonic processes it controls include body axis patterning, cell fate specification, cell proliferation, and cell migration. These processes are necessary for proper formation of important tissues including bone, heart, and muscle. Its role in embryonic development was discovered when genetic mutations in proteins in the Wnt pathway produced abnormal fruit fly embryos. Later research found that the genes responsible for these abnormalities also influenced breast cancer development in mice. The clinical importance of this pathway has been demonstrated by mutations that lead to a variety of diseases, including breast and prostate cancer, glioblastoma, type 2 diabetes, and others. Background and etymology: the discovery of Wnt signaling was influenced by research on oncogenic retroviruses. In 1982, Roel Nuss and Harold Varmus infected mice with mouse mammary tumor virus in order to mutate mouse genes to see which genes could cause breast tumors when mutated. They identified a new mouse proto-oncogene that they named Nt1. It was determined that Nt1 has a high degree of conservation across several species, including humans and Drosophila. Its presence in Drosophila melanogaster led researchers to discover in 1987 that the Nt1 gene in Drosophila was actually the already known and characterized Drosophila gene known as wingless. Since previous research by Christiane Narwan-Quarter SSLEIN Vollard and Eric Weishaus had already established the function of WG as a segment polarity gene involved in the formation of the body axis during embryonic development, researchers determined that the mammalian Nt1 discovered in mice is also involved in embryonic development. Since Nt1's discovery in 1982, continued research would lead to the discovery of further genes related to Nt1. However, since all those genes had not been identified in the same manner as Nt1, it quickly became clear that the Nt gene nomenclature, or naming system, was no longer adequate. Thus, the Nt wingless family was renamed the Wnt family and Nt1 became Wnt1. The name Wnt was chosen because it is a combination, or portmanteau, of Nt and WG and stands for wingless related integration site. Proteins. The Wnt proteins are a diverse family of secreted lipid-modified signaling glycoproteins that are 350–400 amino acids in length. The type of lipid modification that occurs on these proteins is palmitoylation of cysteines in a conserved pattern of 23–24 cysteine residues. Palmitoylation is necessary because it initiates targeting of the Wnt protein to the plasma membrane for secretion, and it allows the Wnt protein to bind its receptor due to the covalent attachment of fatty acids. Wnt proteins also undergo glycosylation, which attaches a carbohydrate in order to ensure proper secretion. In Wnt signaling, these proteins act as ligands to activate the different Wnt pathways via paracrine and autocrine routes. These proteins are also highly conserved across species. They can be found in mice, humans, Xenopus, zebrafish, Drosophila, and many others. Mechanism. Foundation. Wnt signaling begins when one of the Wnt proteins binds the N-terminal extracellular cysteine-rich domain of a Frizzled family receptor. These receptors span the plasma membrane 7 times and constitute a distinct family of G-protein-coupled receptors. However, to facilitate Wnt signaling, co-receptors may also be required alongside the interaction between the Wnt protein and FZ receptor. Examples include lipoprotein receptor-related protein 5/6, receptor tyrosine kinase, and ROR2. Upon activation of the receptor, a signal is sent to the phosphoprotein Dishevelled, which is located in the cytoplasm. This signal is transmitted via a direct interaction between FZ and DSH. DSH proteins are present in all organisms and they all share the following highly conserved protein domains: an amino-terminal DIX domain, a central PDZ domain, and a carboxy-terminal DEP domain. These different domains are important because after DSH, the Wnt signal can branch off into several different pathways and each pathway interacts with a different combination of the 3 domains. Canonical and non-canonical pathways. The 3 best characterized Wnt signaling pathways are the canonical Wnt pathway, the non-canonical planar cell polarity pathway, and the non-canonical Wnt calcium pathway. As their names suggest, these pathways belong to one of 2 categories, canonical or non-canonical. The difference between the categories is that a canonical pathway involves the protein I2-catenin while a non-canonical pathway operates independently of it. The canonical Wnt pathway. The canonical Wnt pathway is the Wnt pathway that causes an accumulation of I2-catenin in the cytoplasm and its eventual translocation into the nucleus to act as a transcriptional coactivator of transcription factors that belong to the TCFLEF family. Without Wnt signaling, the I2-catenin would not accumulate in the cytoplasm since a destruction complex would normally degrade it. This destruction complex includes the following proteins: auxin, adenomatosis polyposis coli, protein phosphatase 2A, glycogen synthase kinase 3 and casein kinase 1I+or-. It degrades I2-catenin by targeting it for ubiquitination, which subsequently sends it to the proteasome to be digested. However, as soon as Wnt binds FZ and LRP5/6, the destruction complex function becomes disrupted. This is due to Wnt causing the translocation of the negative Wnt regulator, auxin, and the destruction complex to the plasma membrane. Phosphorylation by other proteins in the destruction complex subsequently binds auxin to the cytoplasmic tail of LRP5/6. Auxin becomes dephosphorylated and its stability and levels are decreased. DSH then becomes activated via phosphorylation and its DIX and PDZ domains inhibit the GSK3 activity of the destruction complex. This allows I2-catenin to accumulate and localize to the nucleus and subsequently induce a cellular response via gene transduction alongside the TCFLEF transcription factors. The non-canonical planar cell polarity pathway. The non-canonical planar cell polarity pathway is one of the 2 Wnt pathways that does not involve I2-catenin. It does not use LRP5/6 as its co-receptor and is thought to use NRH1, RIK, PTK7, or ROR2. As in the canonical Wnt pathway, the Pcp pathway is activated via the binding of Wnt to FZ and its co-receptor. The receptor then recruits DSH, which uses its PDZ and EEP domains to form a complex with Dishevelled associated activator of morphogenesis 1. DAM1 then activates the small G protein Rho through a guanine exchange factor. Rho activates Rho-associated kinase, which is one of the major regulators of the cytoskeleton. DSH also forms a complex with RAC1 and mediates profilin binding to actin. RAC1 activates JNK and can also lead to actin polymerization. Profilin binding to actin can result in restructuring of the cytoskeleton and gastrulation. The non-canonical Wnt calcium pathway. The non-canonical Wnt calcium pathway is the other Wnt pathway that does not stimulate accumulation of I2-catenin. Its role is to help regulate calcium release from the endoplasmic reticulum in order to control intracellular calcium levels. Like other Wnt pathways, upon ligand binding, the activated FZ receptor directly interacts with DSH and activates specific DSH protein domains. The domains involved in Wnt calcium signaling are the PDZ and EEP domains. However, unlike other Wnt pathways, the FZ receptor also directly interfaces with a trimeric G protein. This co-stimulation of DSH and the G protein can lead to the activation of either PLC or cGMP-specific PDE. If PLC is activated, the plasma membrane component PIP2 is cleaved into DAG and IP3. When IP3 binds its receptor on the ER, calcium is released. Increased concentrations of calcium and DAG can activate CDC42 through PKC. CDC42 is an important regulator of ventral patterning. Increased calcium also activates calcineurin and CAMCA. CAMCA induces activation of the transcription factor NFAT, which regulates cell adhesion, migration, and tissue separation. Calcineurin activates TAK1 and NLK kinase, which can interfere with TCF-I-catenin signaling in the canonical Wnt pathway. However, if PDE is activated, calcium release from the ER is inhibited. PDE mediates this through the inhibition of package, which subsequently causes the inhibition of calcium release. Integrated, convergent, Wnt signaling pathway. The binary distinction of canonical and non-canonical Wnt signaling pathways has come under scrutiny and an integrated Wnt pathway has been proposed. Some evidence for this was found for one Wnt ligand. Very recently, evidence for a convergent Wnt signaling pathway, that shows integrated activation of Wnt CAR2+ and Wnt-I-catenin signaling, for multiple Wnt ligands, was described in mammalian cell lines. Other pathways, along with the pathways described above, Wnt signaling also regulates a number of other signaling pathways that have not been as extensively elucidated. One such pathway includes the interaction between Wnt and GSK3. During cell growth, Wnt can inhibit GSK3 in order to activate mTOR in the absence of I2 catenin. However, Wnt can also serve as a negative regulator of mTOR via activation of the tumor suppressor TSC2, which is upregulated via DSH and GSK3 interaction. During myogenesis, Wnt uses pKa and CREB to activate the genes MyoD and MYF5. Wnt has also been seen to act in conjunction with RIK and SRC to allow for regulation of neuron repulsion during axonal guidance. Wnt regulates gastrulation when CK1 serves as an inhibitor of RAP1-GTPase in order to modulate the cytoskeleton during gastrulation. Further regulation of gastrulation is achieved when Wnt uses ROR2 along with the CDC42 and JNK pathway to regulate the expression of PAPC. DSH can also interact with APKC, PAR3, PAR6, and LGL in order to control cell polarity and microtubule cytoskeleton development. While these pathways overlap with components associated with PCP and Wnt calcium signaling, they are considered distinct pathways because they produce entirely different responses. Regulation: in order to ensure proper functioning, Wnt signaling is constantly regulated at several points along its signaling pathways. For instance, as previously mentioned, Wnt proteins are palmitoylated. The protein porcupine mediates this palmitoylation process, which means that it helps regulate when the Wnt ligand is secreted by determining when it is fully formed. Secretion of Wnt protein is further controlled with proteins such as Nt and evenness interrupted and complexes such as the retromer complex. Upon secretion, the ligand can also be prevented from reaching its receptor through the binding of certain proteins such as the stabilizers DALI and glypican-3, which inhibit diffusion. At the FZ receptor, the binding of proteins other than Wnt can antagonize signaling. Specific antagonists include IFKF, Wnt inhibitory factor 1, secreted Frizzled-related proteins, Cerberus, FRZB, WISE, and SOST. All of these constitute inhibitors of Wnt signaling. However, other molecules have been shown to act as activators as well. For example, norrin and ospondin 2 have been shown to activate Wnt signaling in the absence of Wnt ligand. Interactions between different Wnt signaling pathways also regulate Wnt signaling. As previously mentioned, the Wnt calcium pathway can inhibit TCFI2-catenin in order to prevent canonical Wnt pathway signaling. Induced cell responses, embryonic development, Wnt signaling plays a critical role in the embryonic development of a variety of organisms. It is detected in both vertebrates and invertebrates, including humans, frogs, zebrafish, C. elegans, Drosophila, and numerous others. It was first known to be involved in the segment polarity of Drosophila, where it helps to establish anterior and posterior polarities. However, it has since then been implicated in numerous other developmental processes. As its function in Drosophila suggests, it plays a key role in body axis formation, particularly the formation of the anteroposterior and subventral axes. It is also involved in the induction of cell differentiation to prompt formation of important organs such as the lungs and ovaries. Wnt further ensures the development of these specific tissues through proper regulation of cell proliferation and migration. These are just a few Wnt functions, but they demonstrate that the numerous functions of Wnt signaling can be divided into one of the following categories: axis patterning, cell fate specification, cell proliferation, and cell migration. Axis patterning: in early embryonic development, the formation of the primary body axis is a crucial step in establishing the overall body plan of each particular organism. The different axes include the anteroposterior axis, subventral axis, and right-left axis. Wnt signaling can be implicated in the formation of the anteroposterior and subventral axes. Wnt signaling activity in anterior-posterior development can be seen in several organisms including mammals, fish, and frogs. In mammals, the primitive streak and other surrounding tissues produce the morphogenic compounds WNTS, BMPs, FGFs, nodal, and retinoic acid to establish the posterior region during late gastrula. These proteins form concentration gradients, and the areas of their highest concentration establish the posterior region and the areas of their lowest concentration indicate the anterior region. In fish and frogs, I2-catenin produced by canonical Wnt signaling causes the formation of organizing centers, which, alongside BMPs, elicits posterior formation. Wnt involvement in the subventral axis formation can be seen in the activity of the formation of the Spman organizer, which establishes the dorsal region. Canonical Wnt signaling production I2-catenin induces the formation of this organizer via the activation of the genes twin and CMYE. Similarly, in avian gastrulation, cells of the collus sickle express different mesodermal marker genes that allow for the differential movement of cells during the formation of the primitive streak. Wnt signaling activated by FGFs is responsible for this movement. Wnt signaling is also involved in the axis formation of specific body parts and organ systems that are a part of later development. In vertebrates, sonic hedgehog and Wnt morphogenetic signaling gradients establish the subventral axis of the central nervous system during neural tube axial patterning. High Wnt signaling establishes the dorsal region while high SHH signaling indicates in the ventral region. Wnt is also involved in the dorsal ventral formation of the central nervous system through its involvement in axon guidance. Wnt proteins guide the axons of the spinal cord in an anterior-posterior direction. Wnt is also involved in the formation of the limb dorsal ventral axis. Specifically, Wnt7 helps produce the dorsal patterning of the developing limb. Cell fate specification, or cell differentiation, is a cellular process where undifferentiated cells can become a more specialized cell type. Wnt signaling induces differentiation of pluripotent stem cells into mesoderm and endoderm progenitor cells. These progenitor cells are then further induced to differentiate into more specific cell types such as endothelial, cardiac, and vascular smooth muscle lineages. Wnt signaling can also induce blood formation from stem cells. Specifically, Wnt3 leads to mesoderm-committed cells with hematopoietic potential. Wnt1 has also been shown to antagonize neural differentiation and is a major factor in self-renewal of neural stem cells. This allows for regeneration of nervous system cells, which is further evidence of a role in promoting neural stem cell proliferation. Wnt signaling has also been shown to be involved in germ cell determination, gut tissue specification, hair follicle development, lung tissue development, trunk neural crest cell differentiation, nephron development, ovary development, and sex determination. Cell proliferation: in order to have the mass differentiation of cells needed to form the specified cell tissues of different organisms, a proliferation, or cell growth, of embryonic stem cells must take place. This process is mediated through canonical Wnt signaling, which increases nuclear and cytoplasmic level of I2 catenin. Increased levels of I2 catenin can initiate transcriptional activation of proteins such as cyclin D1 and c-Myc, which control the G1 to S phase transition in the cell cycle. Entry into the S phase causes DNA replication and ultimately mitosis, which are responsible for cell proliferation. This increase in proliferation is directly paired with cell differentiation because as the stem cells proliferate, they are differentiated into the specific tissues that are induced to become. This allows for overall growth and development of specific tissue systems during embryonic development. This is apparent in systems such as the circulatory system where Wnt3 leads to proliferation and expansion of hematopoietic stem cells needed for red blood cell formation. Cell migration. Cell migration during embryonic development allows for the establishment of body axes, tissue formation, limb induction, and several other processes. Wnt signaling helps mediate this process, particularly during convergent extension. Research has shown that signaling from both the Wnt-Pcp pathway and canonical Wnt pathway is required for proper convergent extension during gastrulation. Convergent extension is further regulated by the Wnt calcium pathway, which blocks convergent extension when activated. Wnt signaling also induces cell migration in later stages of development through the control of the migration behavior of neuroblasts, neural crest cells, myocytes, and tracheal cells. Wnt signaling is also involved in another key migration process known as the epithelial mesenchymal transition. This process is what allows epithelial cells to transform into mesenchymal cells so that they are no longer held in place at the laminin. It involves a downregulation of cadherins so that cells can detach from laminin and migrate. Wnt signaling is an inducer of Wnt, particularly in mammary development. Insulin sensitivity. Insulin is a peptide hormone involved in glucose homeostasis within certain organisms. Specifically, it leads to upregulation of glucose transporters in the cell membrane in order to increase glucose uptake from the bloodstream. This process is partially mediated by activation of Wnt2 catenin signaling, which can increase a cell's sensitivity to insulin. In particular, Wnt10B is a Wnt-protein shown to increase this sensitivity in skeletal muscle cells. Clinical implications: cancer. Ever since its initial discovery, Wnt signaling has had an association with cancer. When Wnt1 was discovered, it was first identified as a proto-oncogene in a mouse model for breast cancer. The fact that Wnt1 is a homologue of Wg shows that it is involved in embryonic development, which often calls for rapid cell division and migration. Misregulation of these processes can cause unwanted cell growth and movement, which can lead to tumor development. Activity of the canonical Wnt pathway is involved in the development of benign and malignant breast tumors. Its presence is indicated with elevated levels of I2 catenin in the nucleus and/or cytoplasm, which can be detected with immunohistochemical staining and Western blotting. Increased I-squared catenin expression is strongly correlated with poor prognosis in breast cancer patients. This accumulation may be due to several factors such as mutations in I-squared catenin, deficiencies in the I-squared catenin destruction complex, most frequently by mutations in structurally disordered regions of APC, overexpression of Wnt ligands, loss of inhibitors, and/or decreased activity of regulatory pathways. Breast tumors have also been seen to metastasize due to Wnt involvement in the epithelial mesenchymal transition. Research looking at metastasis of basal-like breast cancer to the lungs has shown that repression of Wnt I-squared catenin signaling can prevent Mt, which can inhibit metastasis. Wnt signaling has also been implicated in the development of more than just breast-type cancers. Changes in CTNNB1 expression, which is the gene that encodes I-squared catenin, can be measured in not just breast cancer, but also colorectal cancer, melanoma, prostate cancer, lung cancer, and several other cancer types. Increased expression of Wnt ligand proteins such as Wnt1, Wnt2, and Wnt7a have been observed in the development of glioblastoma, oesophageal cancer, and ovarian cancer respectively. Other proteins known to cause multiple types of cancer in the absence of proper functioning include ROR1, ROR2, SFRP4, Wnt5a, WIF1, and those of the TCF-LEF family. Type 2 diabetes, or diabetes mellitus type 2, is a common disease that causes reduced insulin secretion and increased insulin resistance in the periphery. It results in increased blood glucose levels, or hyperglycemia, which can be fatal if left untreated. Since Wnt signaling is involved in insulin sensitivity, malfunctioning of its pathway could be involved in the development of type 2 diabetes. Overexpression of Wnt5b, for instance, may increase susceptibility to type 2 diabetes due to its role in adipogenesis, or fat production, since obesity and type 2 diabetes have a high comorbidity. Wnt signaling is also a strong activator of mitochondrial biogenesis. This leads to increased production of reactive oxygen species known to cause DNA and cellular damage. This ROS-induced damage is significant because it can cause the development of acute hepatic insulin resistance, or injury-induced insulin resistance. Mutations in Wnt signaling-associated transcription factors, such as TCF7L2, are also linked to increased susceptibility to type 2 diabetes. See also, signal transduction, morphogenesis, developmental biology, embryogenesis, cancer, catenin, GSK3, FRZB, wingless localization element 3, baldness treatments, GPA177, external links, mouse Wnt proteins from signaling gateway molecule pages, mouse frizzled proteins from signaling gateway molecule pages, the Wnt homepage from NUS Lab, Stanford, Wnt pathways, their relationship, disease, and therapies by healthvalue.net, Drosophila Wnt pathway from KEGG, mouse Wnt pathway from KEGG, Humani plus or minus Wnt pathway from KEGG, Homo sapiens Wnt pathway from KEGG, video of gray hair reversal, NewtPath, a curated resource of signal transduction pathways in humans, Wnt proteins. At the U.S. National Library of Medicine medical subject headings, Wnt game, references.